Yeah, good evening, everyone. I welcome you all to the second live problem solving session for week 2 of NPTEL Bioinformatics Algorithms and Applications course. So, in this session, we'll be trying to understand about the summary of the last week lectures, that is week 2 lectures. So, which is mostly about understanding the micromolecules, which are blending blocks of lives, followed by introduction to sequence analysis such as sequence alignment and other topics, followed by I have some sample problems. And today we'll also be seeing how a dot matrix is constructed using a collab notebook, and how a dot matrix varies for different DNA sequences. Yeah, to start with the introduction to biological macromolecules. So firstly, macromolecules are massive molecules that are formed through polymerization of small unit called monomers. So this term macromolecules was first coined by Hermann Staudinger in 1920. So biological macromolecules, so these are large and complex molecules that are essential for various biological processes in living organisms. There are four main types of biological molecules in mammalian systems. So they are proteins, carbohydrates, nucleic acids, and lipids. Proteins are fundamental macromolecules with diverse roles in cellular process. They are composed of amino acid side chains which fold into a specific three-dimensional shapes. Proteins serve as enzymes catalyzing chemical reactions, or they serve as structural components maintaining cellular integrity, or they can also function as a transporters, facilitating the molecular movement, and also function as signaling molecules, enabling the communication within the cell. So some examples include enzymes that drive metabolic pathways, hemoglobin responsible for oxygen transport in blood, and antibodies defending against infection. So next we have carbohydrates, which is another crucial macromolecule, function as both an energy source and structural support in cells. So carbohydrates are polymers of carbon, hydrogen and oxygen and exist in various forms including monosaccharides, disaccharides and polysaccharides. Examples such as glucose acts like primary energy source while cellulose serves as a structural component in plant cell walls. Next we have lipids which are characterized by their hydrophobic nature and play essential roles in energy storage, insulation and formation of cell membranes or can call as a plasma membranes. Examples such as triglycerides acts as stored fuel reserve. Phospholipids contribute to the structural integrity of cell membranes. Cholesterol, which is a steroid, plays a regulative role in cellular process. Lastly, we have nucleic acids, namely known as DNA and RNA, so which are critical macromolecules involved in the genetic information storage and transmission. So nucleic acids are structured as polymer of nucleotides and they consist of a pentose sugar, a phosphate group and a nitrogenous base. Uh, the widely known one is DNA with its double helical structure serves as the primary repository of hereditary information, while RNA usually single-stranded and acts as a messenger in protein synthesis. Together, these macromolecules, the four classes of macromolecules, intricately contribute to the structure, function, and regulation of cells, organisms, ensuring their vitality of living systems. So now we know the individual functions of these macromolecules. The interconnectedness of these macromolecules is also important. Proteins are crucial for the synthesis of lipids and carbohydrates, while nucleic acids store genetic information which guides the synthesis of RNA followed by the translation of these RNA into a functional proteins. So therefore, carbohydrates, nucleic acids, proteins and lipids collectively contribute to the integrated process of a biological system. So I have a question for you. So those who are attending my session for the first time, I'll just have a brief introduction. Meanwhile, you can provide your answers through the chat box. So my name is Ramakrishna Reddy. So I'm a PMRF scholar working in IIT Madras. So I'll be taking Monday slot 6 to 8 live problem solving session for NPTEL Bioinformatics Applications and Algorithms course. So this is a week 2 lecture. This mostly deals about the recap to the week 2 lectures followed by the some hands-on problem solving sessions on week 2 related to week 2 assignment. Currently on screen you can see the question, which is which macromolecule stores hereditary information in cells, whether proteins, lipids, nucleic acids or carbohydrates. I'll wait for a minute for others to answer. Okay, thank you for the answers. So here I have a question regarding the calculating average hydrogen bond energy. Why do we consider bonding energy between Ga and Ct when we just have two hydrogen bonds between A and T and three hydrogen bonds between G and C only? Should the H bonding energies be calculated with the complementary sequence. Yeah, actually, in reality, the H bonding energies has been calculated only between G and C when we consider intra-strand, interstrand. So when we consider the interstrand, the other concept of G and CT comes, the bonding between them, and also these are just informational values that Sir has discussed about the average hydrogen bond energies. So even similarly, we have other concepts coming up in the previous structure-based analysis session. Then why we need to consider GA and CT will be discussed in week 4 or week 5 lectures. So it's mostly because there is some uh, energy values that are required to go forward with some energetic or mechanistic calculations. So why G and CT average hydrogen bond energies are also considered in bioinformatics. So here the question is, which macromolecule stores hereditary information in cells? So it is
Nucleic acid.
nucleic acids. Next I have one more. So this is about what is the basic structure of nucleic acids contain, or other way nucleic acids are polymers of
Nucleotides.
yeah. So nucleotides are the basic structural. So first we'll start with the proteins. So as we all know, nucleic acids such as DNA and RNA are structured polymers of nucleotides. So this contains a pentose sugar, phosphate group, and a nitrogenous base. Starting with the proteins. So first amino acids which are called as building blocks of proteins. So these are organic compounds containing basic amino group, carboxyl group. So proteins consist of long chain of amino acids forming both peptides and proteins. Among the 20 amino acids, some are synthesized by the body, which are called as non-essential amino acids, while others must be obtained to the diet, which are called as essential amino acids. So 20 existing amino acids has been classified into non-essential and essential amino acids. Non-essential amino acids include alanine, asparagine, arginine, aspartic acid, glutamic acid, cysteine, proline, glycine, glutamine, tyrosine, and we have some remaining amino acids which are essential amino acids, and these must be opted into diet. The common structural feature of amino acid include amino group, that is NH3 group, and a carboxylate group, and a hydrogen bonded to the same carbon atom. The distinguishing feature between these 20 amino acids is the side chain R group. Each amino acid has 4 groups, of which 3 groups are same, that is alpha carbon amino group, COH group, and hydrogen atom are common, and they varies in their side chain atom. So this image shows the classification of amino acids. Properties such as charge, hydrophilicity, and hydrophobicity, size, and functional group influence this classification. Water soluble proteins bury hydrophobic residues, exposing hydrophilic side chains. And whereas integral membranes are inside out versions of the water soluble proteins, which have exposed hydrophobic amino acids and anchoring them into the lipid bilayer. So, these amino acids exhibit diverse properties based on their side chain structures. So, first is aliphatic amino acids. So, these amino acids have aliphatic side chains which are linear and branched. So, examples include glycine, alanine, valine, leucine, isoleucine, and proline. Then we have small amino acids. These amino acids are relatively smaller side chains. Examples include glycine, alanine, serine, threonine, cysteine, and valine. Then we have polar amino acids. So these polar amino acids have hydrophilic side chains that can form hydrogen bonds. Then we have two classes of charged amino acid, that is positively charged or basic amino acids, or negatively charged or acidic amino acids. So the positively charged amino acids include lysine, arginine, and histidine, whereas negatively charged amino acids include aspartic acid and glutamic acid. So overall, if you see, many of these amino acids fall under hydrophobic amino acids class, which have non-polar side chains and are typically repelled by water. And finally, we have aromatic amino acids, which have aromatic ring structures in their side chain. So these include phenylalanine, tyrosine, and tryptophan. Understanding these categories helps in predicting the behavior of amino acids in various biological contexts, including their roles in protein structure, function, and interaction. Special properties of these amino acids include cysteine can form covalent disulfide bonds. Proline forms cycle in the polypeptide backbone itself. It forms a cyclic structure. Glycine is more flexible than other amino acids, and various hydrophobicity scales exist for these amino acid residues. So here I have one more question. So what sets amino acids apart from each other? So whether the 20 different types of are amino acid because of the difference in hydrogen bonding, or difference in their side chain or R group, or difference in their amino group, or due to difference in their carboxylate group. So amino acids, as we discussed, they differ in their R group. So the answer is B, side chain or R group. Which amino acids in the given following list are considered as charged and considered as basic? So as we discussed, we have two classes of charged amino acids, that is positively charged and negatively charged. So here I wanted to know which of the given following amino acids are considered as charged and basic. So amino acids such as lysine, arginine, and histidine are charged and considered basic because they carry a positive charge on their side chain. So just moving on to the next session, I just wanted to know how many of these current participants have submitted their assignment one. I know the deadline is till Feb 5th. Just you can reply with a yes in the chat box or hands raise. Okay, thank you, thank you. Okay, thank you for your answers. Let's move ahead. Starting with the structural diversity of proteins. Proteins exhibit diverse shapes and molecular weights, and they are characterized as globular and fibrous proteins. So this protein three-dimensional shape, what we are talking, is crucial for function, and this any changes in the this three-dimensional structure may lead to uh, protein denaturation, or sometimes it may lead to diseases. So this protein shape is based on amino acid composition, as it influences protein properties, determining whether they are acidic, basic, polar, or non-polar. Native conformation, that is the natural 3D shape of a protein, is maintained by the primary sequence of a protein. Further, it has been classified into secondary, tertiary, and quaternary levels. So at the primary level, proteins are defined by their unique sequence of amino acids forming a linear chain, which is similar to beads on a chain. So this sequence, like a biological code, sets a foundation for other levels of the structure. Moving to the secondary structure, so this has local folding patterns emerging. So these local folding patterns include helices, sheets within the chain. So these folds give the protein more intricate shapes resembling spirals or flat structures. Then we have tertiary structure. So this involves the overall 3D arrangement of the entire protein, a complex interpret dictated by the interaction between distant amino acids. We can visualize this tertiary structure as a unique 3D shape of the entire uh, beads on a necklace, essential for the protein's function. So this is completely a functional form of a protein. Finally, we have a quaternary structure which introduces collaboration, has multiple protein chains or subunits come together to form a fully functional multi-subunit complex. The widely known example is hemoglobin, with its four subunits working in tandem to transport oxygen. So beyond this, we have quinary structure, which delves into the interaction between proteins and other molecules. So the major types of interactions that one can see with a protein is protein interacting with other protein interactions, protein interacting with nucleic acids, that is protein DNA interactions or protein RNA interactions. Then we have protein interacting with carbohydrates, that is protein carbohydrate interaction. So these quinary structures adding an extra layer of complexity to their roles within the biological systems. From primary to quinary structure, each level intricately contributes to the protein's overall structure and function, highlighting the remarkable complexity of these essential biomolecules. So therefore, understanding protein structure and function provides insights into the dynamic and vital roles they play in the complexity of living organism. So here I would like to pose a question for you, which is what emerges at the secondary level structure of a protein. So during secondary level, what do we observe? Overall 3D arrangement we observe, or multiple subunit collaboration we observe, local folding patterns we observe, and quinary structure interactions we observe. we see local folding patterns which leads to the formation of alpha helical or beta sheet structures. Next, to start with the yeah, before moving on to here, I just wanted to discuss about small thing about protein classes and function. Protein exhibit diverse roles based on their structure, environment, where they reside or fold. So broadly telling globular proteins and membrane proteins. Globular proteins are typically soluble and many function as enzymes. Their role is involved in various cellular processes and often exhibit enzymatic activities. Then we have membrane proteins. These proteins are integral part of the cell membrane which span the cellular membrane several times, and these membrane proteins, based on secondary structure classification, they are broadly classified as alpha helical membrane proteins and beta barrel membrane proteins. And when we observe the pattern, the folding pattern of these membrane proteins, which is completely different from globular protein, has their functional environment include highly hydrophobic lipid molecules. So these membrane proteins have several functions like transporting molecules in and outside the cell. They also serve as receptors for cell-cell communication and also enzymes in various biological processes. So this understanding the distinct roles and structures of these protein classes is crucial for comprehending the complexity of cellular process and diverse functions they perform in living organism. So now to start with the bioinformatics sequence alignment, which is used for unveiling the relationships and functions of sequence and with the sequence. So in bioinformatics, sequence alignment is a crucial technique for arranging DNA, RNA or protein sequences to unveil regions of similarity that indicate potential functional, structural or evolutionary relationship between the sequences. So this process involves representing aligned sequence of nucleotide or amino acid residues within a matrix with gaps introduced as introduced between residues to align identical or similar regions in successive columns. Sequence alignment is important for various purposes in bioinformatics and molecular biology. Some key applications include it helps to identify the regions of similarity that may indicate functional similarity. That is, proteins or nucleotides with similar sequences often perform similar biological function. That is, similarities in sequences can imply similarities in 3D structures, then similarities in 3D structures may imply similarity in biological functions. By aligning sequences, that is, by comparing two sequences, one can infer the evolutionary relationship between different species or genes. So the conserved regions in alignment mostly suggest the common ancestry. So sequence alignments are generally used to annotate the genes and identify various genetic elements within the genome. This is essential for understanding the functional components of DNA. So sometimes sequence alignment extend beyond just comparing biological sequences, that is like calculating the distance caused between the strings in natural language or financial data. So comparing sequence and identifying relation. To compare sequence effectively and aligning conserved and non-conserved residues across all the sequences necessary. So this alignment helps to identify the locations of insertions or deletions, shortly known as indels, since the divergence of the common ancestor. Phylogenetic program leverage these residues to find out the distance relation between the sequences. So in general, when one perform a sequence alignment, when perform a sequence alignment, either three types of conditions are observed: match, mismatch, or a gap, introduction of gap. So match indicate no change since divergence. Mismatch signifies a substitution since divergence. So substitution can be regarded as a mutation. Then gap indicates one residue in one sequence and no residue in other. So this implies an insertion or deletion since divergence. So the application of sequence alignment in bioinformatics include DNA sequence assembly. So aligning DNA sequence data aids in assembling larger contiguous DNA sequences. Then alignment in protein helps to identify crucial regions like active sites in proteins. Alignment also implies relatedness between sequences, whether due to shared ancestry or like orthologs, paralogs, or common genetic origins. So this sequence alignment can also be further classified as pairwise sequence alignment and multiple sequence alignment. So pairwise sequence alignment methods are used for finding the best match alignments between two sequences, any two query sequences. So the common methods include dot matrix technique, dynamic programming techniques such as Needle Manunch and Smith Waterman algorithms, and word based methods. While efficient, these methods encounter challenges in highly repetitive sequences of low information content, particularly when repetition patterns differ between the sequences. So today mostly I'll be dealing with the dot matrix method of the sequence alignment. So before that, I have a question for you, which is what is the significance of identifying locations of insertions or deletions in the sequence alignment? Yeah. So the significance of identifying locations of insertion deletion, that is shortly indels, in a sequence alignment is inferring relationships since a common ancestors divergence. Next I have discussion about dot matrix. So this dot matrix approach is qualitatively and conceptually straightforward method for aligning sequences. So although this method is considered as time consuming for large scale analysis, this technique is particularly useful in visually identifying certain sequence features such as indels, repeats, inverted repeats without any noise introduced. To construct a dot matrix plot, two sequences are represented along the axis of a plot. So we can tell axis of a two-dimensional plot. So a dot is placed, like a star represents a dot here. A dot is placed at any position where the characters in the corresponding columns, like characters in the x column, x axis, and characters in the y axis match. So this results in a recurrence plot, and the size and intensity of dots may vary based on the degree of similarity between the characters. So in closely related sequences, the dot plots will appear as a single line along the main diagonal. However, dot plots have some drawbacks as an information display technique. So dot plots are only limited for comparing two sequences. Further, these dot plots can also uh, serve as serve to assess the repetitives within a single sequence. When a sequence is plotted against itself, regions sharing significant similarities appear as the lines of the main diagonal. So this phenomenon is observed when a protein comprises multiple sequence structural domains. So as I discussed, to further delve into dot matrix, it's a graphical representation used to compare two nucleotides or a protein sequences, highlighting similarities, differences, and structural features between them. So how a dot matrix is made? First is sequence preparation. So you start with two sequences that you want to compare, say sequence A and sequence B. So these sequences could only be nucleotides or proteins. Then followed by matrix construction. So this matrix constructions where the sequence are plotted on the x-axis and the y-axis. A dot is placed in the matrix when nucleotide or amino acid from sequence A and sequence B at corresponding positions match. For example, if nucleotide at position 1 in sequence A matches with the uh, nucleotide at position 1 in sequence B, a dot is placed at position 1 comma 1 in the matrix. If they don't match, no dot is placed. Often there is a nowadays we have seen a concept called window size which is employed. So this matrix often employs a window size of 1 to 5 nucleotides or amino acids in case where window slides along the both sequences to find the local regions of similarity. A threshold score can also be applied to define whether it's a match or is significant. So this is an example of a simple dot plot. So if the aligned sequences can be observed along the diagonal, so where, yeah, can anyone tell me why there is a empty box where my cursor is hovering? What does this significant when we compare two sequences?
No alignment in the sequence that doesn't exist in sequence 1 or sequence 2, like T to G, where the gap is there. I mean, one gap is there, T to G, T on the y axis and G on the x axis. That box, no T to G exists in sequence 1 and 2, so there is a gap.
Yeah, okay. Yeah, thank you for your answer. Yeah, it's a mismatch or it's a gap in the alignment. So the common features that we are observed in a dot matrix is similarity, dissimilarity, insertions, shortly known as indels, mutations, inversions, palindromes. So regions with many matching dots form a diagonal lines in the dot matrix, indicating the areas of similarity between the two sequences. Long and continuous diagonal lines suggest that highly similar uh, two sequences are highly similar. Then we have dissimilarity or a mismatch. Areas with no dots are scattered dots in a diagonal across the either sides of a diagonal line represent regions where the sequences differ significantly. So the absence of a diagonal line shows that no substantial similarity exists in that particular region. Further, we have insertions deletions. If one sequence contains extra nucleotides not present in other, this will appear as a gaps in the dot matrix. These regions are marked by broken or disjoint diagonal lines.
Sir,
Similarly, deletion. Yes, please.
sir, how we have put the dot in this graph? Suppose in the first sequence A is there, and in the second sequence T is there, but we have put the put the dot instead. They are dissimilar to each other.
Yeah, I think there is a shift in the one column is missing. So a dot is placed. Yeah, I'll just correct this. Sorry, thank you for this correction. I'll correct this in the code thing. I'll show you during the code. Yeah, followed by that, we have mutations, just point mutations where one nucleotide is substituted for another will cause a smaller deviation or mismatches in the diagonal line. So now I'll be sharing a code, a link in the chat box, which is a Google Colab. You can directly copy paste. I'll wait. So you can copy this link that I have shared, copy, then go to your browser, paste it. So you will be redirected to a Colab notebook. Every one of you, can you see this Colab notebook or able to reach here?
Yes, sir.
So here initially I have taken two sequences. This is just a string of letters. So the string here tells the quick brown fox jumps over the lazy brown dog, has sequence 1, and the same thing I have taken as sequence 2. And now I wanted to see how the dot matrix between two similar sequences look. So I'll run this. Yeah, you can see a run button here, and if you click, the corresponding cell will get executed. So this is the dot matrix that corresponds to the quick brown fox jumps over the lazy brown dog. Since two sequences are highly similar, we can see a continuous diagonal line that is present. So here a dot is placed because in the first letter we have T, then here in the sequence 1 and sequence 2 we have T in the first place, and also we have T here in the second the. So a dot is placed here followed by here. Then in second place of sequence 1 we have H. So here H we have at second position in the sequence 2. So a dot is placed in the second position here. So further I want to see the other scenarios. So the other scenarios is a insertion happening. So in a new sequence 2, I have, instead of jumps, I have added a 5 letter size string kkk. So this identifies, this tells the jumps has been replaced, substituted with some other letters. So here now I wanted to see how this plot got changed. So to deselect the initial sequence, just add this hashtag symbol in front of the second line sequence 2, and just remove the hashtag symbol in front of third line sequence 2 and click on run. Now we can see a gap has been introduced here. So this gap is because of the substitutions happened for the word jump. Now I am selecting the other sequence too. So can anyone see and tell what is the difference between the sequence 1 and sequence 2, which are showing in a white color? So what is the difference between the sequence one? Okay, I'll just
The word jumps is missing.
so this tells, yeah, the word jump is missing, so this is a deletion happening in the sequence. So now I want to see what happens during the deletion scenario, or in other case insertion in sequence 1 or deletion in sequence 2. Yeah, one can see there is a shift in the diagonal. Previously we have seen a gap in the diagonal, now there is a shift in the diagonal. So the shift in the diagonal identifies insertions or deletions in the sequence. Yes, please.
Sir, this indicates mutations also, right? Information regarding mutations,
Yes,
insertion, deletion.
yeah, insertions or deletions are also considered as part of mutation. So we have a substitution under mutations concept. We have substitutions, insertions, and deletions.
Okay, sir. Thank you, sir.
Yeah, so here I have one question. Along with this, okay, just change the sequence. Yeah, so along with the main diagonal region, we have two other small diagonals that are parallel to our main diagonal. So what does this tell? So based on the sequence alignment you see, so one diagonal corresponds to the and other diagonal corresponds to brown.
Duplication.
Yes.
Missing words are there, like quick in the first case and lazy in the second.
No, actually I'm asking about the parallel lines that corresponds to the and brown. Yeah, they are copies. So here, if we take a sequence, the sequence itself has the copies of repetitive words, and brown is also repeated. So because of this, if there is any repeats, we see them as parallel lines to the main center diagonal. Yeah, yes, repeated sequences. Now that we have taken some a string, now a similar thing we can do with a sequence. So here I have taken a sequence. So sequences, this is a complete similar identical sequence. So when I run, this is the dot matrix you observe. So this dot matrix looks little clumps. So this is due to the noise or 2D visualization of. So this is one of the major drawback of the dot matrices. Followed by that, we have concept of inversions. So inversions are special case in dot matrix when a part of a sequence or complete sequence is in reverse orientation. So the diagonal lines will appear mirrored. So now we have seen diagonal from leftmost corner to the right top. So in inverted case, we should see the mirrored version of the diagonal. So here I have taken a sequence 2 which is an inverted, complete inverted sequence of sequence 1. I'll try to run it. So this is how inverted or inversions that are observed in a dot matrix. So, okay. This is the actual dot matrix uh, telling the highly similarity or a highly identical sequence followed by having repeated sequences within itself. So when I change the sequence to its inverted sequence, that is, this sequence is sequence 2 is inverted version of sequence 1. So this is how you see now the diagonal lines run from left top to the right bottom. That is, the diagonal lines will be appeared mirrored. Then we have other concept, that is palindrome. So a palindrome sequence is a region that reads same forward and backwards. So in a dot matrix, palindromic sequences will form a diagonal line that symmetrically mirrors itself across the central axis of the matrix. So you can see a 90 degrees perpendicular line. This is a X shaped occurrences that happens. So here I have taken some example for visualization purpose. So this is how a palindromic sequence. So this X indicates, the aligning of this X indicates presence of a palindromic sequence. That is, sequence reads same from the start and same from the end. That is A B C D E E D C B A, same A B C D E E C B A. So this gives the presence of a palindromic sequence. So these are the some special cases, and I have also discussed about the window size. So generally the window size in dot matrices are considered small, that is 1 to 2 nucleotides. A smaller window size increases the resolution of the dot matrix, allowing a finer details. So this window size in dot matrix analysis plays a crucial role in determining the resolution and sensitivity of the comparison between two sequences. The window size refers to the number of nucleotides considered at a time during the comparison process. Changing the window size can have significant impact on how well different features of the sequences are detected and how the results are interpreted. So what happens when we select a smaller window size? So when you choose a smaller window size, you increase the sensitivity of the dot matrix to smaller variations like mutations, SNP single nucleotide polymorphisms, and mirrored structural differences. Larger windows, on the other hand, might overlook the minor differences since they would require a larger stretch of mismatching nucleotides to form a dot, potentially ignoring a smaller local variations. So actually there's a concept called balancing between the detail and general pattern. Like smaller windows gives you more detailed information highlighting very local variations and differences, but they might make the matrix harder to interpret due to the much finer details. Larger windows are better for identifying the general patterns or large scale similarities or differences between the sequence, but might miss out on fine grained mutations or changes. So observing a dot matrix here, can you mention what is the window size that I have used here?
1.
Yeah, I'll wait for others also to answer. Others can use chat box to mention what is the window size that I have used in the collab note. And why it is 1, how it is 1. So thank you for the answer. In this whole alignment, I have considered one nucleotide at a time. So here the window size is 1. So let me move back. When I save this, I'll update this dot plot and I'll post. So now that the explanation of dot matrix is done, I have a question, which is, what is the primary purpose of the dot matrix approach in the sequence alignment?
Sir,
Yes, please.
sir, can we get this PDF for later revision?
Yeah, so all these lectures along with the live recording of this lecture followed by the material, that is PPT of the slides and a link for the collab notebook, I'll be posting in the Google Drive.
Sir, from the first day live class also?
Yeah, already I have posted first day live class.
Okay, sir, okay.
Yeah, sorry, I forgot to ask. Everybody are able to follow the collab notebook, or anybody is having issues in the collab notebook, or I can show it again how to access the collab notebook.
Sir, kindly show it.
Yeah, so I have given a link. Okay, again I'll share the link. So I have shared the link. You just click on the link, copy, then go to your browser, paste the link, enter. So this is the code. So in order to run SL is a box, like this is a cell. In order to run this part of the code, you can see there is a play button or run button here. So when you hover it, it tells run cell. So you just click on it. Okay. So below the cell, the result will be updated.
Thank you, sir.
Yeah. So this collab note is made in Python. So it's not only restricted the coding, or this dot plot can also be done in other coding languages. So one can select his own language of interest or expertise and can do this. Since I am comfortable with Python, I have done this in Python. Okay, let's move on to the question. Yeah, so generally the primary purpose of a dot matrix in sequence alignment is to visualize the regions of similarity. I have one more. So how many sequences are generally represented in a dot matrix plot, and how they are represented? Yeah, uh, just for easy visualization, I have made the borders for the matrix. Actually, dot plot is a matrix. It's a plot which has x and y axis, not rows and columns. Just to visualize and for back tracing, I have added borders to the plot. This is not a dot, how a dot matrix look. This is how a dot matrix look. It's a plot, scatter plot. Yeah, so here along the x and y axis of the matrix, can anyone mention me what will be the disadvantage of dot matrix, or when the dot matrix becomes clueless or can't help us?
Sir, when we have more than two sequences,
Okay, other than that, okay, I have only two sequences. Then, in what scenarios dot matrix cannot help us in visualizing the similarities or dissimilarities?
we can't compare the sequences among more than two species.
Yeah, okay,
No similarity at all.
okay. What if,
Lots of repetitions.
okay, what if I tell, is it easy to visualize a sequences length of 100 or sequences length of 2000, which is a simpler dot plot, which is made with
100.
so what if I increase 2000, 3000, and all, the complexity of the plot increases
Yes, sir.
and the resolution of the plot decreases. So the major limitation of dot plot is number of sequences it can handle, followed by the length of the sequences. So we have other, so to overcome these, we have some other concepts called dynamic programming. So dynamic programming is an algorithmic technique used for solving optimization problems by breaking them down into simpler overlapping problems. So in bioinformatics, dynamic programming is widely employed for sequence alignment, including Smith-Waterman and Edelman-Wunsch algorithms, that is global alignment and local alignment algos. So these algorithms optimize local and global sequence alignments respectively by finding the best alignment with maximum similarity score. So the first one is Needleman-Wunsch. It is a pairwise global alignment algorithm. That is, it constructs a matrix to find the optimal alignment of two sequences by maximizing a similar score. So given two sequences, it finds the optimal alignment which has a maximum similarity score that is possible for alignment of these two sequences. So this is a global sequence alignment method. So this global sequence alignment method aligns from starting of a sequence to the end of a sequence. It considers whole for the alignment purpose. So this alignment process is done by assigning scores for matches, mismatches, and gaps, determining the overall optimal similarity between the sequences. Then we have Smith-Waterman. So this is for local sequence alignment. Smith-Waterman is also kind of similar alignment to Needleman-Wunsch, but it has some changes. So this focuses on local similarities within the sequence. It won't align from one end of a sequence to other end. It's try to find the local similarities. Followed by that, we have BLAST. The full form of BLAST stands for Basic Local Alignment Search Tool. So it is widely used program in bioinformatics. We have different variations of BLAST, that is BLAST and BLAST P. So generally the BLAST is used for comparing biological sequences. It searches for a local sequence alignment between query sequence. Query sequence is your input sequence and a database of sequence. BLAST employs heuristic approach to effectively find the significant matches, making it suitable for large-scale sequence databases. So the major purpose of a BLAST is, given an input sequence, we can try to identify the homologous sequence in a database for a given query. It uses heuristic to accelerate the search, including word seeding and extension method. And BLAST also employs a scoring system based on matches, mismatches, and gaps, and provides a list of significant matches with statistical measures such as e-values, indicating the probability of finding the match by chance. So this is just a basic introduction to these dynamic programming methods. Next week, uh, solving how a Needleman and a Smith-Waterman algorithms are constructed, followed by the demo session of the BLAST I will be doing. Now I have some questions. So this is the problem solving session here on. So what is the primary purpose of Needleman-Wunsch algorithm in bioinformatics? Whether the Needleman-Wunsch algorithm is used for
So option B.
yeah, it is option B for global sequence alignment. So which algorithm is specifically designed for local sequence alignment? So these algorithms are named after the person who has developed them. It's a Smith Waterman. So what does BLAST stand in for bioinformatics? Yeah. So BLAST stands for Basic Local Alignment Search Tool. So we'll start with the assignment question. So what is the one element that proteins have but carbohydrates and lipids do not have? I'll wait for a minute for others to answer. Yeah, so nitrogen is the one element that proteins have in their amino group but carbohydrates and lipids do not have. So these are the basic structures. So nucleic acids have N group, proteins have N, but carbohydrates and lipids doesn't have the nitrogen group. So the second question is, what level of protein structure is primarily influenced by hydrogen bonding? Yeah, the derivatives of these biomolecules have nitrogen group. So I was mentioning basic subunits of carbohydrates and lipids doesn't have a nitrogen group. Okay, I see some answers telling secondary structure. So what are the secondary structures that we widely hear or see in protein structures? Names of the secondary structures.
Alpha helix and beta pleated sheets.
Secondary structures are primarily influenced by the hydrogen bonding pattern among themselves or with the other secondary structures. So the primarily observed secondary protein structures are beta pleated sheets and alpha helix. So alpha helix has hydrogen bond. Sorry, alpha helix have hydrogen bond within the chain, and beta sheets have with the other beta sheets. The third question is, what level of structure allows for the hemoglobin to have 4 identical subunits? It's a quaternary structure which includes subunits, tertiary subunits coming together to form a functional form, quaternary form. So this is the level of protein structures. So primary level is sequence, then we have secondary structure, followed by tertiary structure, then the quaternary structure. So quaternary structure of a protein consists more than one amino acid chain or protein chain. So given a condition of sickle cell anemia includes mutation to valine. So in this scenario, which type of hemoglobin is apparently more polar and insoluble in water? So in this sickle cell anemia, the glutamic acid at 6th position on hemoglobin is mutated to valine. So the normal hemoglobin has glutamic acid at 6th position. So what type of hemoglobin is this? Yeah, so due to the presence of charged and polar amino acid here, the normal hemoglobin is more polar and soluble. And next, which type of hemoglobin is more non-polar and insoluble, and why? Yeah, so now the new sequence, that is sickle cell hemoglobin, has valine at 6th position. So valine is an hydrophobic amino acid, that is non-polar amino acid. So this makes this hemoglobin, which is more non-polar and insoluble. So here now I have question 5, which is proteins which are embedded in the outer membrane are. Yeah, read all the options carefully. I'm asking proteins which are embedded in the outer membrane are, mostly observed in the outer membrane are. So the options have given us globular proteins, fibrous proteins, alpha helical membrane proteins, and beta barrel membrane proteins. So here I have mentioned about two types of membrane proteins, alpha helical membrane proteins and beta viral membrane proteins. So these two class of membrane proteins are mainly differed by their membrane spanning secondary structures and also have the difference in their folding patterns and functional patterns. So the widely observed ones in outer membrane are beta viral membrane proteins. So this is how beta sheets arrange themselves in a barrel-like structure and are embedded into the outer membrane. And each membrane protein also has extracellular and periplasm domain also, which is similar to the globular protein domains. So this is how a structure of beta viral protein embedded in outer membrane looks like. And mostly they function as channel proteins. So the main properties of channel proteins are whether they are used for gating, selective ion conduction, gative and selective ion conduction, yeah, they are used for both gating and selective ion conduction. So what is a comprehensive database of enzymes? Whether string is a comprehensive database of enzymes, Brenda is a comprehensive database of enzymes, DDBJ or UniProt. So Brenda is a comprehensive database. Next, eighth question is, in the system of nomenclature, which one of the following enzyme occupies the first position? So the enzyme commission, that is EC system, classifies enzymes into 6 major classes based on the type of reaction they catalyzed. Each enzyme is assigned an EC number in the format of EC X dot Y dot Z dot W. So where the first digit, it is 4 digits. The first digit gives enzyme class, that is reaction type. Second digit gives subclass, reaction specificity. Third digit gives specific group involved in the reaction. Fourth digit is serial number of the enzyme within that subclass. So first we have reductases. They function in catalysis of oxidation reduction reactions where sorry, electrons are transferred between molecules. So these oxidoreductases commonly involve NAD plus, NADP plus, and FADS electron acceptors. So some example of oxidoreductases are alcohol dehydrogenase and cytochrome C oxidase, which converts alcohol dehydrogenase converts ethanol to acetaldehyde involved in alcohol metabolism, whereas cytochrome C oxidase plays a key role in cellular respiration, that is in electron transport chain. Then we have second class is transferases, that is EC2. So they function for transferring the functional groups from one molecule to the other molecule. So here we have kinases, transaminases, aminotransferases, so which transfer amino groups in amino acid metabolism. Then we have hydrolases, which function in the catalysis of hydrolysis reaction, that is breaking bonds using water. So these hydrolases are highly observed in digestive enzymes or degradative enzymes. Examples such as amylases, proteases, and lipases falls under the category of hydrolases. Then we have lyases, which breaks the chemical bond without using water or redox reaction, and these lyases involved reaction often form double bonds or cyclic compounds. That lyases include decarboxylases, aldolases, etc. Then we have isomerases. Similarly, as the name suggests, they also involved in catalysis of isomerization reactions, which involve rearranging molecules without changing the molecular formula. Then we have ligases. So ligases are used to join two molecules together using ATP or any other energy source. So these are has essential role for DNA repair, replication, and biosynthetic pathway analysis experiments. So along with that, now I wanted to introduce the concept called scoring matrices. Scoring matrices are essential tools in bioinformatics used to measure the similarity between biological sequences. These matrices assign numerical scores to match, mismatch, and gaps in the sequence alignment. So we have broadly two types of scoring matrices, that is substitution scoring matrices and distance based scoring matrices. So substitution scoring matrices are used in alignment related scenarios, whereas distance matrix is used in phylogenetic tree constructions and all. That is, distance based matrices are used to measure the evolutionary distance between the sequences. So the widely known one is PAM, which stands for point accepted mutation. So a PAM matrix is a tool used in bioinformatics and computational biology to quantify the evolutionary distance between the homologous pairs, protein or DNA sequences. It helps in assessing the likelihood of mutations occurring at a specific positions within these sequences. So this PAM was introduced by Margaret Dayhoff in 1970s. So the basic idea behind a PAM model is to quantify the probability of a particular amino acid substitution over time, assuming a constant rate of evolution is happening. And generally the unit of measurement is PAM, so which represent 1% change in amino acid sequence. So here the PAM number represents evolutionary distance. So one PAM unit means 1% of amino acid mutated on average. Higher PAM value corresponds to longer evolutionary divergence. Means, for example, if we consider PAM 250, means that on average each amino acid has changed 2.5 times over the evolutionary time. And how do we use PAM matrices? So when we are working with the closely related proteins, it is better to go with the lower PAM number, that is PAM 10, PAM 1, PAM 100. When we are working with the distance related proteins, it is better to go with the PAM higher number, PAM 250 and others. So these PAM matrices are commonly used in sequence alignment algorithms such as BLAST and FASTA. So now I have the question, which is the central hub for collection of functional information on proteins with accurate, consistent, and rich annotation is. asking database for proteins. Yeah, so this week I couldn't discuss the answers of the week 1 because still the deadline is not yet over. So next week I'll try to discuss the solutions to the assignment 1 questions. Yeah, so UniProt, which is a unified database for proteins and its annotation information, and it is also a primary resource for protein information including sequence, structure and other annotation, functional annotation information. So UniProt KB is made up of two large sequence collections. That is, one is a manually annotated and reviewed, and other one is automatically annotated and reviewed. So these two versions of UniProt TB are called as. For this question, you can refer to the UniProt database and see what are the two major listings that are available. Okay, SwissProt, the other one. So I'll tell. So go to uniprot.org, type any protein name. For example, I am using insulin. Search. So you can see the UniProt entries. The status. So we have reviewed under SwissProt, unreviewed under TRNBL. So SwissProt in UniProt has manually annotated and reviewed sequences, and TRMBL has automatically annotated and not reviewed sequences. Similar description. Reviewed is SwissProt, unreviewed is TRMBL. Then for the alignment, the match score is 3, mismatch score is -1, and the gap score is -1. So what is the net score? So consider the match score as 3, mismatch score as -1, and the gap score is -1. Okay, let's move on to. So this is the given alignment. So let's slide score. So the first condition A is a match, so I am writing 3. Next condition, it is C and G, it's a mismatch, so -1. Then again it's a match, G is matching. Then a gap has been introduced after G, then it is -1. Further, next there is a mismatch, that is -1, gap, -1 and a match. So overall this is 9 minus which equals to 5. So the answer is 5. This is how alignment is scored for a similarity score. This 5 is the alignment score or a similarity score of this alignment. So I have some other alignment. So in this alignment the match score is 4, gap score is -2, so I'll wait for a minute for others to solve. So for a match case, a score has 4 has been allotted. For a gap, score of -2. So this is overall 12 minus 5, which is equals to 7. Yeah, so I have some basic question. What is the average molecular weight of an amino acid residue in a protein? In Daltons? So this is 110. Yeah, now I have a last question for the day, which of the following is the correct order of increasing hydrophobicity for a given amino acids? So the given amino acids are alanine, isoleucine, valine and phenylalanine. So I have given a justification for a practice purpose. In exam, these justifications won't be given. So based on the justification given, select the option. here the first three options are same, or the answer A, B, C, anything is fine. Here the justification given is least hydrophobic among the given amino acid due to its small methyl side chain is alanine, which has only CH3. Then we have valine, which is more hydrophobic than alanine due to its isopropyl side chain. Followed by that we have isoleucine, it has bulkier alkyl side chain. Then we have phenylalanine, which has more hydrophobic nonpolar benzyl group. Yeah, so these are the tasks that I wanted you people to get familiarized before the next class. So this will be easier for us to discuss about uniprot and Needleman and Smith-Waterman algorithms before. So that's all for today. If you have any other questions, I can take the questions. If not, you are free to leave the class.
Excuse me, I didn't get that PAM 250, like in a for this phylogenetic tree analysis. So, if a protein sequence, how can a amino acid sequence be more than 100%? Like, how can it be 250% vary, the variation?
See, actually, we have different versions of the PAM, like I just mentioned. So, for example, if you take PAM 250, means that on average each amino acid has changed 2.5 times over the evolution. That is, it could be changed into different other versions. On an average, 2.5 times. Like for example, alanine to valine is one mutation, valine to other is the second mutation, and third mutation. On an average change, this 2.5 is defined as an average change. But one PAM unit means 1% amino acids mutate on average.
So, 250 means all the 100% amino acids have been mutated 2.5 times.
Yeah, for example, if you take a chain of 100 amino acids, the number 250 means in these sequences along the evolutionary chain, these 100 residues have 250 mutations. They have gone overcome the 250 mutation. Like 250 types of mutations has been observed in these 100 sequences. So now they are distantly related sequences. If less number of mutations are happening among these 100, they are closely related sequence in that sense. It's not like between two sequences. This throughout the evolutionary change, first evolutionary change could relate in 100 mutations, then again remaining 100 followed by 50. It's mean. Just an observation. 250 mutations are observed per 100 sequences in PAM 250 compared sequences.
Okay, thank you.
If no other questions, thank you for attending. Guys are free to leave.
Thank you, sir.